Chapter 2 deals with biochemistry basics. So atoms are the smallest units of elements, and they're pure substances that make up ordinary matter. So atoms are comprised of protons, which are positive, neutrons that don't have a charge, and electrons, which are negatively charged particles. So some terms to know, guys. Ions, those are charged atoms that have an unequal number of protons and electrons. Cations are atoms that have lost electrons, therefore they have an overall positive charge. And anions have gained electrons, and therefore they have an overall negative charge. So later in the semester, guys, we'll be talking about oxidation and reduction, and that's where you're going to have a loss or a gain of electrons. So some different types of chemicals to know. Acids would be those that add hydrogen ions, and a hydrogen ion would be a H+. And bases add hydroxide, and hydroxide are OH-. So that's can be used then when we start to talk about pH. So the H+ and O- determine the overall acidity or basic properties of a solution. So pH then is the negative log of the concentration. Anytime you see brackets, that means concentration of hydrogen. So the scale then describes how basic, that would be down here, the higher numbers, or acidic, the lower numbers down here, that a chemical would be. So pure water would be neutral. So that would then have a pH of 7. If you have a basic or alkaline solution, the pH is going to be greater than 7, and acidic solutions will then be less than 7. Now in some experiments that are done, we actually had a pH indicator called phenol red. And in acidic environments, the phenol red is actually going to turn this yellow color. That's going to be important when we talk about fermentation in the laboratory. Now, different types of molecules, fats in particular, can be what we call hydrophilic or hydrophobic. And hydrophilic are water-loving, and they'll dissolve in water. Hydrophobic are water-fearing, therefore they do not readily dissolve in water. So think of oil. Oil is hydrophobic. Now, molecules can also be what we call amphipathic. So these have both hydrophobic and hydrophilic. So if we take a look then at our lipid bilayer, the top part here would be our hydrophilic part, and down here the fatty acids, these guys here, those are hydrophobic. Now we'll be talking about energy production this semester, and two terms I'd like you to know are exergonic and endergonic. Our exergonic reactions occur when reactions release more energy than is spent to start the reaction. The products have a lower final energy than the reactants. In endergonic reactions, these occur when reactions use more energy than is released. So the products have a higher final energy than the reactants. And we'll be talking about all of these throughout this semester, but these are our four main classes of biomolecules: carbohydrates, nucleic acids, proteins, and lipids. And I'll just go through a couple of quick things on those. So carbohydrates are energy sources, structural molecules. They can help cells adhere, communicate, and sense things in the environment. The one I'd like you guys to know is in red, and that's peptidoglycan. And the reason I'd like you to know that is every bacteria has peptidoglycan. So only bacteria have this. It's part of the bacterial cell wall. Capsules are another important type of carbohydrate that cells have. Basically, it's a smooth type of glycocalyx. It helps the cells adhere, and it also protects them from our immune system. Lipids are fats, oils, waxes, etc. And there are a lot of these that we'll discuss this semester. So one of the structural components of tuberculosis, guys, is that it has a waxy coat on the outside. So we'll discuss why that's a problem when you're trying to treat a patient with TB, because it does have that waxy coat. We also have things known as steroids and sterols, and again, these are important for different types of hormones and also for cholesterol production. So lipids function in energy as energy sources and cell structure and mediate cell signaling as well. So please take a screenshot of this particular slide, guys, upload it to the Dropbox. So one, as I mentioned before, mycolic acid is a waxy type of substance found in tuberculosis and leprosy. Okay? It increases the pathogenicity of TB and leprosy, which means that their disease-causing ability is ramped up. So when you think of the mycolic acid, guys, think of wax, like candle wax, and you know how hard that is. So what you're trying to do when you're treating the patient then that has TB is get rid of that wax so the rest of the bacteria that's underneath it can actually be killed. So nucleic acids, we'll talk about DNA and RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And this just gives you an idea. We have a nucleotide, that's a structure that has a phosphate sugar and a base. The sugars then would be ribose for RNA, deoxyribose for DNA. So please know the four bases that are in DNA are adenine, guanine, cytosine, and thymine. Adenine, guanine, cytosine, and thymine. When we look at RNA, please know we still have adenine, guanine, and cytosine, but we have uracil instead of thymine. So if you ever come across a nucleic acid sequence, if you see uracil, you know that you're looking at RNA. Now, proteins, please know, are made of amino acids. We're going to discuss later in the semester protein synthesis. Please know that each amino acid has three parts. It has an amine group, which is NH3, carboxyl group, COOH, and side groups that we call R groups. So this is where the variety of different amino acids comes from, is that R group. That side chain is what makes them all different.